We live in a visual world where the amount of reflected light, the luminance, can change by more than 10,000 times throughout the day. Because of these large light fluctuations, the white and black parts of this subway sign can reflect 1,000 times more light at noon than at night. How can our perception of black and white be so reliable throughout the day? It is currently believed that cortical neurons signal luminance contrast independently of light intensity. That is, the luminance response function of neurons in visual cortex is the same when the maximum luminance is 100 candles per square meter than when it is 100 times higher. However, if this was true, a bright sky should look like this. It should look very dark because the luminance of the clouds is much lower than the luminance of the sun. To address this puzzle, we measure the luminance response functions of neurons in primary visual cortex. We drove cortical responses from the on pathway with white stimuli, shown in red, and from the off pathway with black stimuli, shown in blue. We presented the black and white stimuli at different luminance ranges, spanning from a maximum luminance of 300 to 1000 candles per square meter. To our surprise, we found that the shape of the luminance response function changed with both the luminance range and the black-white stimulus polarity. Increasing the amount of light had opposite effects on the on and off cortical functions. It shifted the luminance contrast that generated 50% of the maximum response, the L50, to the right in off neurons and to the left in on neurons. Making the image brighter increased the off L50 on L50 ratio. This effect could be demonstrated in both visual cortex and natural images. Image brightness increased the separation between the distributions of dark and light contrast in nature. It also increased the separation between on and off contrast response functions in visual cortex. The amount of light is continuously changing in our visual world. Our work demonstrates that image brightness changes our sensitivity to light and dark contrast to efficiently extract information from natural scenes.